Now we are going to proceed with the Julie Stout presentation.
Hello, this is Julie Stout. I'm here today to present to you a talk on hippocampal-dependent memory and Huntington's disease. I'm at Monash University in Australia at the Turner Institute for Brain and Mental Health. I want to thank the chairs of this session, Jaime Kulisevsky and Saul Martinez Horta, for the invitation to speak with you today. Let's start at the clinic. People with Huntington's disease, they don't usually come in with the first thing they say that they have a problem with their memory. But when you discuss their memory with them, they'll tell you that they spend ages going over things in their head, what needs to be done, but they still forget, or that they can't remember what they did yesterday. And then someone reminds them and they'll say, oh yeah. Or they can't remember what their daughter's activities are for each week unless they write them down. If they don't write them down, they forget what the activities are. So people with HD do have memory complaints. And as a neuropsychologist, our first thinking about these memory complaints tends to be that they're caused by damage in the frontostriatothalamic brain circuits, especially at the level of the striatum. We believe that their inefficiency in learning information and retrieving that information is the biggest part of their memory impairment. But when you look more closely, you see that they also have problems with storage. And we can see this on word list learning tasks, for example. So this is a study from the Predict HD project. This is a study from 2007 published from my lab. And we looked at the ability of people in the pre-manifest period to be able to encode new words from a word list, to store that information and to retrieve it. We have 4 groups that are CAG expanded here. CL0s are the furthest from onset. CL3s are the closest to estimated onset. And what we can see here is the healthy non-CAG expanded group. And we have observed here problems in encoding that start well into the pre-manifest period, perhaps worse in the group that's closest to onset. We see significant declines in the ability, or we see significantly worse performance in the ability to store the information as based on this word list memory testing in the group that's closest to onset, and perhaps that begins a bit before then. And then in the pre-manifest group, we also see evidence of problems with retrieval. This is about the recall of information, and it's about the ability to systematically search and retrieve information. So it's this storage type of memory that we think of as being hippocampally dependent, whereas we think of retrieval as being more related to striatofrontal brain circuits. Now, there is evidence of hippocampal atrophy and also hippocampal pathology in people with Huntington's disease. The hippocampal atrophy is shown on MRI. It's shown clearly in people in the early HD phase in a few studies, also in a couple of other studies in the late pre-manifest period is also observable. We also know that hippocampal subfields, that there are specific hippocampal subfields that are more affected in HD, particularly the Spargo 1993 paper points at hippocampal CA1, which is highlighted as a brain area that's important in cognitive neuroscience research. This is a brain area that's thought to be important for linking together spatial and temporal information in memory processes. So CA1 is quite a key area for episodic memory in that regard. Episodic memory, which is about, when things happened and what happened and where. This just shows you the part of the hippocampus that's most affected in HD. It's the thread region here. This is shown in a coronal slice. This is shown in an axial slice. You can see it kind of blown up here. So it's the red part, which is hippocampal CA1. Other areas are also probably affected, but CA1 has had some attention. Now in this talk, I'm going to talk about 2 studies from my group that are evidence of hippocampal-dependent memory impairment in HD. And I want to say that these studies are also bolstered by a handful of studies from other groups as well. And in particular, I want to point out to you a couple of elegant studies coming from Roger Barker's group at University of Cambridge that I have highlighted here. So the first type of hippocampal-dependent memory I want to talk about is autobiographical memory. Autobiographical memory are our memories and recollections of personally experienced events and occurrences that we link to a specific time and place. And autobiographical memory is known to be affected in Alzheimer's disease and frontotemporal dementia, some other forms of cortical damage, where people really have profound inability to remember things from their past. Autobiographical memory is typically tested using an interview format. The one we use is called the autobiographical memory, the autobiographical interview. Participants are interviewed and asked to recall autobiographical events from 5 periods of their lives. We record what they say, we transcribe it, we segment the memories part into informational details, and we code those informational details using qualitative methods, looking at internal details, which are details that are related to episodic memory, I'll illustrate in a moment, and external details, which are not. So here's an example of an autobiographical memory. I'm just going to read a few sentences from it as examples. First, an example of something that is an autobiographical memory that is episodic because it's linked to some specific thing that happened, specific time and place. The person says, I remember being the first one to arrive because I was kind of excited. I ordered a specialty cocktail that tasted fruity. So these are sort of 2 events, and one of them is an autobiographical memory event that's episodic, that has a thought or emotion component. I was kind of excited because I was the first one to arrive, or when I was the first one to arrive. And here's another one. Another event was, and I ordered a specialty cocktail that tasted fruity. So that's another event. It's a perceptual, has a perceptual element because it refers to taste. Okay, these are contrasted to another type of event where the person says, sushi is my favorite food. That is not an episodic event. It doesn't have any element of time or place. It's just about the person's knowledge about themselves. So we would call that a semantic autobiographical memory. And what we're interested in for hippocampal dependent memory are those memories linked to time or place, like these that I highlighted here, but not so much these that are just the semantic nature. So we studied 24 healthy controls and 26 people with the CAG expansion, 14 manifest, 14 pre-manifest. We studied them under 2 conditions. One was a free recall condition where we just asked them to remember and tell us back these events from these different parts of their lives. And the other was a probed recall condition where we gave them a memory aid, which was we probed them to try and remember certain events that had occurred previously in their lives. This helps people to remember things when they're when they're inefficient in their retrieval processes, like people with HD are. And what we found, whether it was in the free recall condition or the probed recall condition, was that people with HD recalled fewer autobiographical memory events. They're shown here in this dark gray compared to the healthy controls in white. They have fewer spatiotemporal details. That's a really strong sign of a hippocampal memory. They have fewer evidence of perceptual memories of these events, fewer emotional memories of these episodic memory events. So people with HD clearly have problems in their autobiographical memory. This is a peri-manifest group. We didn't have enough participants to separate into pre-manifest and healthy controls, but we clearly see this in this kind of early HD sample. Now going to spatial navigation and memory, this is another type of memory that we know is affected in HD from a number of studies, but I'm going to just give you an example of one from our group that did an interesting task. This is the ability that's needed to find a location, and usually in relation to an object. This is a task we call it the virtual house task. It's a computerized task that uses sort of 3D. It's a house that people have to navigate around in by navigating on the computer. It has 8 rooms, and participants are told that there's a dog in the house in one of the rooms and that they should try and find it and then remember where it was found. And what happens is they self direct their navigation through this virtual house for 30 to 45 minutes looking for the dog. And after about 30 minutes, the dog's barking periodically. After about 30 minutes, the dog can be found in one of the rooms that has a table. The dog is under the table and it can be found. But they have to navigate for at least 30 minutes through this house. They can navigate as long as 45 minutes to try and get familiar with the layout. So that's part one, is the self-directed exploration. And then part two is the testing. It has 3 conditions. And I'm going to describe those to you in a few minutes. But before that, I just want to point out that the rooms, these 8 rooms, have objects in the rooms. These are just a sample of 2 of the rooms: chairs up against the wall, bookshelves, table and chairs, and so on, a door. But they also have some objects that are placed in arbitrary locations. For example, a faucet or a tap on the wall, and some just geometric shapes like this one here or this one here. So these objects become part of what we ask them about in a subsequent condition. So after they've navigated through the house, the self-navigation, the first test phase we do is a test of their ability to navigate the house. We ask them to go into the house again and navigate to the room that where they previously found the dog as efficiently as they can. So they self-navigate, and what we do is we count the number of spaces they traversed to get to the point where they found the dog, and also how much time it took. So that's those are a couple of the measures. The second type of measure looks at their ability to remember objects in the locations where they were found before. So again, these are the 2 of the 8 rooms that they see, and these are those same 2 rooms in this object location condition. What we do in this sort of test part of the task is we show them these rooms where these blue boxes replace objects that were in the room before, like in these locations here. So you can see, for example, this location has a tap on the wall. Here it's just a blue box. And what we do is we take them through to these rooms. We show them this blue box and we ask them what object was there previously. And so we can look at how many objects they can recall from their 30 to 45 minutes of navigation around the virtual house. And then we look at the number that they recalled out of a total of up to 23. So these are objects placed in the correct locations. There's a third condition, which I don't have time to talk about today, which is a drawing of the floor plan. So the results of this study, we had 33 people in the control group. We had a pre-manifest group and an early HD group. So the first 2 things we looked at again were the navigation to find the room where the dog was. That in the test part of the task, we found that people with HD were both slower and traversed more spaces. That was only statistically significant in the early HD group, who took 127 seconds and traversed almost 5 spaces on their way to find the dog, where the location where the dog had been found, in comparison to healthy controls, which were much speedier and and navigated to fewer locations on the way. So they were more efficient. But the place that really struck us in terms of these results was really in the object location part of the task, when we asked them to navigate with us through the rooms and to tell us where those blue boxes were placed, what object had appeared there when they were exploring the house. And we found that although the healthy controls could name correct object locations for about 12 and a half out of the 23 possible, people in the pre-manifest group only got an average of 9, and people in the early HD group got an average of about 7.5. And I'll just highlight for you that this healthy control difference between HD, pre-manifest HD and healthy controls, this difference was statistically significant. This is the only one of the tests in this task that was statistically significant in the pre-manifest group, whereas in comparison in the early HD group, all of the variables that we studied were statistically significant. So we think that in this task, it's the object location task, which is something that really looks at the confluence between the location of an object and what that object was, just like we were talking about when I was describing what hippocampal CA1 does. We think that that's showing an early effect here, even in the pre-manifest group. Now, as I said, there's also a spatial, there's a floor plan drawing component to this test as well, which I'm not really highlighting, but the trends were similar, but not as statistically, not as big of an effect. Now, just in coming to the conclusion here, I want to say that we really do know that there are hippocampal memory deficits in Huntington's disease, and we also know that the hippocampus is abnormal in HD. We know that this is clearly present in people who are in the manifest phase of the disease, but it seems to appear in the pre-manifest phase as well, although, of course, the effects are more subtle. But what's the importance of this finding? Well, it's important to think about what episodic memory does. We know from models in healthy controls, for example, Eleanor McGuire and colleagues, that this idea of spatially coherent scenes is important, not just for episodic memory and for spatial navigation, but also in our ability to think about the future and to use our imagination. These are also functions that are really relevant to being able to use spatially coherent scenes, scenes that link places and objects. And this is something that we really need to think a little bit more about in HD. So for clinical purposes, why might this be important? Well, it's possible that for people who have problems, for example, with their autobiographical memory, this autobiographical memory impairment could have an impact on their interpersonal connectivity. One of the ways we relate to people we know is about our shared memories of the past, and it could well be that the inability to share the richness of those past memories in people, in interpersonal relationships of people with HD and their partners, it could be that this reduction in that ability to exchange those memories could have an impact in the way they relate to each other. Another thing to think about is the ideas of Endel Tulving, a very important memory researcher, who really talked about the sense of self, and he talked about this term called autonoetic awareness. And autonoetic awareness is like a person's sense of themselves, who they were in the past, who they are likely to be in the future, who they are now. And this is a very memory-oriented way of thinking about oneself. By knowing oneself from the past, we can know ourselves for the future. And he talks about the fact that when people have these profound inabilities to retrieve memories about themselves, that it creates the sense of just floating in space and not having an anchor to the past or an ability to look to the present, to the future. So the sense of self or autonoetic awareness is something that might well be affected in HD. It's not something obviously people come into the clinic complaining about, but it really could be a part of their experience that we haven't really probed and tried to understand. So we also know that there are gaps in our understanding about how people with HD think about the future. We don't know much about how that might affect their ability to plan. And I can tell you, for example, recently I was sitting around with some colleagues and we were discussing some of this research, and a couple of my colleagues were really struck by it because they were saying that one of the most frustrating things with their Huntington's patients, these are some neurology colleagues of mine, in comparison to their motor neuron disease patients, is that the people with HD were very difficult to engage around planning for their futures, around, putting together an advanced directive, about thinking about financial issues and insurance and what things they might need to do and put things in place for their disease as things progressed. So this might suggest that, if this kind of memory impairment does have an impact on people's ability to think about the future, it might have an impact on their planning, and it might really have some important clinical manifestations for people who have HD in their family. Just finally, I want to have a moment to acknowledge my colleagues, especially Yvette Glickman-Johnston, who I show here. in this picture. She's also written a fabulous paper, by the way, called Spatial Memory and Huntington's Disease, and it's a comparison of human and animal data. I think it's really a great paper, and I think it really lays out this case not only for the hippocampus, but for the ability to use this kind of spatial memory probing approach as a kind of comparative approach for looking across non-human animals to humans, which is really important for us in many of the animal model work, animal model studies that we're all interested in. I also want to acknowledge Anna Carmichael. She was a graduate student in my lab when she conducted that autobiographical memory uh, research project, and finally our research volunteers in Melbourne and across Australia. Thank you. I hope you have a great meeting at the EHDN remote meeting, and I hope to see you next year in person. Bye.